Hey, hi everyone. Good morning. Today we'll continue talking about photosynthesis, the way that plants make ATP from harnessing sunlight. We started with that conversation last week on Friday when we introduced that photosynthesis consists of two different parts. The stage that involves harnessing the sun's energy and converting it into ATP, and then a second part, which is taking some of that ATP and the plant using that to make sugars, right, by taking carbon dioxide from the atmosphere and taking the carbons from those molecules and putting them together with other organic molecules to make different carbohydrates. And so that's the Calvin cycle. So what we'll do today is we'll go over and review some of the concepts that we talked about having to do with ATP production from sunlight, and then we will move on and finish up talking about the Calvin cycle. Okay. So I wanted to point out that from time to time, I'd like to check in and see where we are on the schedule to remind all of you how our semester is progressing. So here we are in week 13. So today's lecture is, again, finishing up photosynthesis, and we'll talk about the Calvin cycle. Take a look at this, right? After this week, next week is the last week of new material, the last week of full lectures. And then the week after that, week 15, is our third exam. And that third exam, wraps up our semester, right? So we don't have a big cumulative final exam in this semester. So we just have our 3 midterm exams. So the third midterm exam will be the same format as the previous 2, and it will cover all the material on this slide from week 10 through week 14. Okay. So I just wanted to give you a heads up because I don't know if it excites you. I'm kind of excited that things kind of wrap up in just a couple more weeks, right? So keep that in mind. So as a result, you will have 2 more quizzes and also 2 more homework assignments, right? And that will finish up those types of assignments for the class. Okay. All right, so let's talk about photosynthesis, and we'll start today where we left off last time. Remember, I told you about how, having grown up on the East Coast, I'm very fond of the sight of trees changing color during the different seasons, right? And in Boston, in the suburbs of Boston, in New England, I used to always love the way the leaves would change from bright green in the summer to oranges and reds and yellows, right, in the fall. Now, I should say, to clarify that, right, when I was a little kid growing up there, to me, it didn't seem all that special, right? Because, typically when you're a kid growing up, you just assume the whole world around you is kind of like what you have around you. And it wasn't until later when I grew up and moved away from the East Coast, moved to California, that I really missed the changing of the seasons. So to this day, I really love the sight of the fall foliage. Here in Hawaii, we don't see a lot of that. We don't see a lot of dramatic changes in color. Right? And there's a reason for that, which we can talk about today. But let's start off with this fundamental question. Why do leaves change color in the fall? Right? And it has to do with the fact that chlorophyll A and B, last lecture, we looked at the structure of these chlorophylls A and B, and they're these very complex organic molecules, right? They have a magnesium ion and a whole series of rings around them with single and double bonds, and then sort of a long branching hydrocarbon chain. That's why I said, in a way, the molecule is almost shaped like a net, right? Like that you would use to catch bugs or catch fish and things at the tide pools, right? And you can imagine a big molecule like that is very complicated and costly for the organism, in this case, the plant, to constantly be making. Right? So these molecules are made, they break down. To constantly make these large, complex chlorophylls requires a lot of resources. Right? If you have a lot of sunlight shining down on you, then it's worth it. right? Because remember, these little net-shaped molecules, what are they catching? They're catching the photons of light. And as the photons hit the electrons in that sort of net-like structure, then the excited electrons start flipping around, single bonds, double bonds, and that energy, right, that's represented by the photon of light hitting the chlorophyll molecule gets transduced into these electrons that are moving around in energy, and they bump into other chlorophyll molecules, and they transmit that energy, eventually starting this electron transport chain, right? That's used by the plant in its chloroplasts to start pumping protons to drive ATP production. Okay? So if you have a lot of sunlight, then it's worth it to make these big complicated molecules. But in places of the world where the sunlight changes in quality and intensity and in availability, depending on what time of the year you're at, maybe there's some times of the year when the amount of quality sunlight really isn't worth it for the plant to be constantly making these chlorophyll A's and B's. So for example, in this cartoon here, let's say in late spring, start of summer, you have this tree, this might be on the East Coast. I'll pretend this is in the suburbs of Boston where I grew up, around Cutler Pond that I used to enjoy walking in. The angle of the sun's light during this time of the year, right, is almost perpendicular to the surface of the planet, right? Because of the angle of the sun and the way the Earth is slightly tilted on its axis. As a result, if you were to choose a small square area, whether this is on the ground, or we can actually pretend this is just a little square area on the surface of a leaf, a green leaf, right? Consider that in this case, you got a lot of photons hitting there, right? So it's worth it for the plant to have lots of chlorophyll, green chlorophyll, ready to receive it. But in the fall, as the Earth's axis is tilting it away from the sun as it's going around, the angle of the sunlight changes. So in order to have the same number of photons being able to get captured, you'd actually have to have a much larger area or surface to receive it. Or I think the better way of thinking about it is that same surface area on a leaf would be receiving far less photons. And in that case, maybe it's no longer worth it for the plant to bother making these big, complicated chlorophyll A's or B's. But hey, at this point, the plant still needs to photosynthesize, right? Because there still is available light. So in this case, other types of pigment molecules become more important. So some of these other types of colored molecules that photosynthesize are molecules like beta carotene and a family of molecules that we call xanthophylls. Okay? So these are accessory pigments that help normally protect cells against intense light that's so intense that it might actually damage the photosynthetic pathway. Right? These beta carotenes, these xanthophylls, what we call accessory pigments, these aren't nearly as complicated as a chlorophyll A or B molecule. So the plant can still produce these, and these can kind of become more important in receiving the light in the fall. So if you look at this figure here, this is just a reminder, right, that chlorophyll A in dark green, chlorophyll B shown in light green. Remember, this figure is showing that as you look across the range of wavelengths of visible light that range from, let's say, 400 nanometers, these would be the sizes of the actual wavelengths contained in the little packets of photons, right, from the sun. Remember, the smaller or shorter the wavelengths, the more energy is contained in that photon, right? The wider the wavelength, like up to, let's say, 700 nanometers on this scale, the lower the energy. Right? And I used a comparison about waves in the ocean, right? When you're close to shore and you're in the splash zone or the crash zone of the waves, right? The waves are much tighter and smaller or narrower, so they have more energy, right? And they can knock you over. Whereas if you wander maybe a couple hundred yards out from shore and you're wading in the water there, those same waves that are coming in, right, their waves, the wavelengths at that point are still much wider. So it ends up just kind of lifting you up and you're sort of bobbing around out there, right? Lower energy at that point. And so on the y-axis, you'll recall that what this is measuring is how well do these different pigment molecules, how sensitive are they to absorbing light in these particular frequencies? And we say chlorophyll A, chlorophyll B, right? They have two sort of ranges that they are sensitive to, right? Sort of in this lower range, so chlorophyll A and B, right? They're able to be very sensitive to light in the blues, and they are also sensitive to light in the oranges and the reds, right? The carotenes, so here are the carotenoids, you can see, they're sort of in the background. They are also sensitive to the blues and the greens, right? But they are not sensitive at all to the yellows, oranges, and reds. So just like we discussed, that's why chlorophyll A and B looks green to us, because they're not sensitive to picking up or absorbing any of the sunlight that's in the green wavelengths. So those green wavelengths of light bounce back off the leaf, and our eyes get to receive them, and we see them as green, right? The carotenoids, you can see, right? They absorb much of the blues and more of the greens, right? But they're not sensitive to the yellows, oranges, and reds. So guess what, right? Those wavelengths of light bounce back from the carotenoids, and we see them as the colors, for example, the fall foliage, right? So as the leaves of the tree from summer into fall begin to lessen or lower the production of chlorophyll A's and B's, right? What we begin to see is more of the carotenoids in the background becoming more visible, right? Because normally those carotenoids, they're always there in the leaf. They're always there in the leaf, but they've just been sort of covered up by so much of the chlorophyll A's and B's. They didn't really appreciate that these oranges and yellows were there all along. So here's the structure of a carotenoid. So this is beta carotene, right? So that's what gives carrots its orange color, right? The carrot, the crunchy root of the carrot is loaded with beta carotene, and it does not absorb yellows and orange light. So that's why it appears orange to us. The structure hopefully looks a little familiar to you, right? This looks a lot like vitamin A, terpene that we studied back when we were looking at different hydrocarbons and different types of lipids, right? So beta carotene is our dietary source of vitamin A, right? You all know you eat carrots because you're told they have vitamin A, right? Technically they have beta carotene, but that beta carotene can then be broken down into vitamin A in our bodies, right? And that's how our bodies then use this particular molecule. All right. So the function of carotenoids and xanthophylls, these so-called accessory pigments, they help out with photosynthesis, right? But importantly, they also act to protect the plant against kind of like getting sunburned, if you want to think about it this way, all right? If you look at certain plants, right, certain plants will change color depending upon how much light they have. So this particular type of succulent or cactus, right, that's maybe used to growing in really sunny, strong sunlight desert areas, in low sun, they might sort of appear kind of greenish. But if they get continuous high sunlight, they turn sort of this nice orangey red. Right? And that's because these pigments such as xanthophylls, they work by absorbing a lot of that extra light that's hitting the plant, right? And kind of acting to prevent too much excitation of the photosynthetic electrons, right? So remember, in this cartoon on the right, we had these large photosystems that are made up of many, many proteins that have things like chlorophyll A, chlorophyll B in them. And you remember they are receiving energy from photons of light, and then the energy is transduced into these double and single bonds flipping around inside the chlorophyll molecules. And as the chlorophyll molecules are kind of bumped up against each other, right, that sort of is a chain of energy being transmitted, right, through resonance transfer. right? Those electrons are not actually leaving a molecule of chlorophyll and going into the next molecule of chlorophyll, right? They're just kind of bumping into each other, sort of like those steel balls in that type of little swinging, what do they call it, Newton's cradle or whatever type of desk toy, right? When you have lots of balls, steel balls in a row, and you swing one down, the balls in the middle, they don't actually move, right? It's the ball at the end that then comes up. Right? So the energy being transmitted through those middle steel balls is resonance transfer. And that's what happens in the photosystems. Right? So eventually, though, the resonance energy from a photon will make its way to the core of the photosystem, where you have that special molecule of chlorophyll known as the P680. And that P680 will cough up an electron. Right? So this one actually gives up an electron because it's excited into a higher energy state, and then that electron might then be booted off and go into the electron transport system. Right? But we mentioned last time that if this P680 chlorophyll is being agitated and losing electrons, those electrons have to be replaced. Right? The moment this P680 loses an electron, it's no longer the same chemical structure anymore. Right? It needs to have another electron or pair of electrons put back in it so that that can be the next pair of electrons that get booted off, right, by all the energy that's coming in into this photosystem. So you can imagine if the sunlight is way too intense and electrons are constantly being booted off of this P680, the plant might get into trouble, right, if it cannot keep up by replenishing the electrons to restore the P680 chlorophyll. So that's where these accessory pigments come into play. So pigments like xanthophyll can actually take the 680's agitated state, right, and absorb some of that energy. And that energy is absorbed so that the 680 doesn't have to give up the electron, right? So it sort of stabilizes the whole reaction here. And then the energy that's absorbed by the xanthophyll, it'll just be converted to heat, and it'll just be released from the plant cell and then from the leaf, right? And as a result, the P680 can remain at its happy, stable state. You know what it's like? You know, in those movies that involve like a nuclear power plant that, you know, goes out of control, and there's like some sort of nuclear incident, right, catastrophe. You know how they always show like the nuclear, what do you call it, the reaction chambers? It's like a swimming pool. That's what it looks like, right? It's like a swimming pool, and you have all the radioactive material down there, and the water is there to kind of keep it cool or whatever. But you have these carbon rods, right? Then in the movies, they can, well, I don't know why I say it's in the movies, because this is how they do it in real life. These carbon rods can be lowered or raised out of the nuclear reaction, because these carbon rods are used to absorb some of the nuclear chain reaction so that the chain reaction doesn't get out of control in the reaction chamber, right? So you can think about it that way. These xanthophylls in the plants evolved to be like that. If you need them, they can absorb a lot of the extra energy to kind of slow down the reaction of the photosynthesis, so it doesn't overload, right, the photosystems of the plant, and so it can keep these important P680 chlorophylls kind of manageable. So that's why in intense sun, right, this cactus over here will upregulate or produce more of these xanthophylls in order to accommodate the fact that it has such strong sun hitting it. So this is not the same technically as getting sunburned, right? I want you to be careful here, right? It's not so much, even though I say it's protect against sunburn, it's not the same reaction that's happening inside of your skin when you get a nice tan in the sun, right? I only use this as an example of how different molecules can be upregulated, right, as sort of a defense against strong sunlight. When you talk about these pigments, these different types of photosynthetic molecules, I think it's important to keep in mind that you have some of them that are very complex, some of them that are much simpler. And this is true not only for things like photosynthetic molecules, but all kinds of molecules that are produced by something like a plant as part of its normal sort of biological functioning, part of its normal life cycles. So in addition to pigments, like colored compounds oftentimes are very complex in order to get all kinds of elaborate colors. Fragrances, so the smell of different flowers that are used to attract things like pollinators, are also often very complicated molecules, right? That's why, the scent of certain flowers are not things that you just smell everywhere around you every single day, right? Because if they were very simple molecules, then you would imagine these simple molecules can be found in all kinds of things, right? But the very, very distinctive, really wonderful perfumey smells of some of plants are very complicated molecules. So that means, right, just like the colors, plants oftentimes have to do a trade-off, right? Do they want to invest their energy in making beautiful colors, right? Or do they want to invest their energy in making wonderful smells? That's why quite often you will have plants that produce flowers that are either very showy and beautiful, right, and very colorful, but maybe don't smell that great, or you have flowers that are very modest, right, simple, small, maybe just white, right, but smell incredible. So here are some examples of some local flowers that I think are quite fragrant, but the reality is when you look at them, right, they don't seem all that showy, right? So certain white plumeria, puakinikini, wonderful example, right? Gardenias, pikake, right? These are all examples of plants that evolved to choose investing their energy into making fragrances, complex scent molecules, right, rather than spending time making a lot of really, really bright, showy colors, right? Okay. So hopefully you understand my point that green leaves that change colors, or any green leaf for that matter, right, actually has these different types of orange and yellow and red pigments already present in the leaf. It's just you don't really see them most of the time because there's a lot more of the green chlorophyll A and B. So that tends to kind of take over. But you can actually do this yourself, a little experiment at home, which is kind of fun, right? So we'll call it homebrew leaf chromatography here. You can take a few green leaves, like these happen to be, I don't know, maple leaves or something like that, right? And you look at these and you're like, oh yeah, these are green. I don't see any oranges or anything in there at all, right? But you can crush them up and soak them in ethanol, in alcohol, like in a coffee cup here for maybe like an hour or so, right? And then all those pigment molecules could then come out of the leaf and be in the alcohol, right? And if you looked at the alcohol at that point, it probably just looked green, right? But this is what you do. You take a couple of paper coffee filters and you cut them into strips, right? And you can kind of dangle them over the coffee cup so that the ends are in the alcohol. And then what happens? Of course, the alcohol will start soaking up into these coffee strips, and it'll start soaking higher and higher, and it'll carry all of these molecules that are kind of dissolved in the alcohol with it. And depending upon how small and simple the molecules are, or how big and complicated these molecules are, they will end up being sort of stuck or trapped by the paper and move at different rates up the filter, right? And what you get is something like this. So after a few minutes, or even longer, as the alcohol has absorbed and gone up the strip, the very complicated, massive chlorophyll A molecules, right, will have only migrated this far along the paper, whereas the much smaller accessory pigments, like the carotenoids, right, they can be carried up much further. Right? So as the paper draws up the ethanol, you can actually separate these different pigment molecules and prove to yourself that all along those wonderful yellow-orange colors were in the leaf to start with. Okay. All right. So what we've been talking about is this first part of photosynthesis. This is the energy transduction part of photosynthesis. Remember, it means the energy from the sun in the form of photons raining down on the surface of the leaf, being absorbed by different photosensitive pigment molecules, and that energy being transduced into electrons, right, that are now moving around through an electron transport chain, right? So again, here is the stack, the thylakoid stack inside the chloroplast of the plant. Here's the light hitting the first of 3 photosystems, right? So this is actually technically photosystem 2 over here. And remember, you'll have that special chlorophyll 680 that's going to cough up a pair of electrons, and those electrons will be the pair of electrons that actually leave that chlorophyll and then get moved through this electron transport system. Right? In order to replenish the 2 electrons coming off of that P680 chlorophyll, right, the plant breaks a water molecule. So that H2O, the 2 electrons and 2 protons are taken off of it. The 2 electrons replace the ones that were booted off the P680, and meanwhile, the 2 protons are just kind of released into the space over here in the lumen. And importantly, what? You get the oxygen left over from the O2 that the plant doesn't care about anymore, right? So it just releases the oxygen, and that is the oxygen on the cartoon on the left here. As water and energy is coming in, oxygen is being released as a waste product. And I think it's wonderful to think that even though it looks very unassuming in this cartoon here, right, this is what you and I depend upon in order to stay alive, right? The oxygen that is being released by this plant into the atmosphere as its waste product becomes the essential oxygen that we need to breathe in to keep our mitochondrial electron transport chain running, right? So these ETS proteins in the thylakoid, they pump protons across. So you build up a very high proton gradient in the thylakoid lumen. And of course, right, these protons are then allowed to flow back downhill along the flow of their gradient, and in doing so power these ATP synthase pumps. And I said, this whole part of the cartoon is very analogous to what happens in the mitochondria, right? But the big difference is, in the mitochondria that's making ATPs, it has different electron transport system proteins, right, that are relying upon electrons being fed into it from those electron carrying baskets, right? Those electron carrying baskets that are dumping their electrons into the mitochondrial electron transport chain. Where did those electrons originally come from? They came from places like the citric acid cycle, right? And the purpose of the citric acid cycle is to fill up those baskets. And what fed into the citric acid cycle? Well, the acetyl-CoA that came originally from the pyruvate from the glycolysis of glucose, right? So that's one way to keep it all together in your head. The mitochondrial electron transport chain, right, is all about a way to take glucose after glycolysis has only gotten a couple crummy ATPs out of it. You can then feed it into the electron transport chain where you generate a lot more electrons, and then those electrons will feed, it'll feed it into the citric acid cycle. The citric acid cycle then generates a whole bunch of electrons that feeds into the mitochondrial electron transport chain. And this similar thing happens, right? Pumps up a bunch of protons. Those protons then power ATP synthase pumps. But in the plant, we are starting from where this whole story starts from, which is the original source of energy coming down to Earth in the form of light, right? And the electrons in this case then are just coming from water, being constantly broken to replenish the source of electrons that are then flowing through these, that are being kind of powered along or shoved along by the energy from the sun, right? All right. So that's part one of photosynthesis. So now you might say, well, I don't get it because the plant has made ATP now. So shouldn't it just be done, right? Isn't that the whole point, is to make ATP so that the other, enzymatic pathways inside of the plant can now use that energy? Yeah, right? But you have to think about this. 2 things. Number one, is the sun always turned on in the sky, right? No, right? Because what is the plant going to do at night? At night, what's going to happen to this whole system up here? It's not going to be working, right? Because at night, for many hours, there is no sun. There are no photons coming in. Right? And so as a result, during the night, the plant is not making ATP. Right? So does that mean at night the plant doesn't need ATP to survive? No, of course not, right? Because it's not like the plant cell dies every night and then is somehow magically reborn in the morning, right? So what does that imply? That implies that there must be a way, as part of this photosynthetic pathway, to be able to already start storing some of that ATP energy that's being made, so that when this energy transduction part of photosynthesis isn't working, like when things are dark, right, that the plant can still have this energy in the form of something else. And what is that something else? Well, you guys know, right? It's glucose, right? That's why photosynthetic life on Earth, the most primitive type of life on Earth, billions of years ago, it was not just about making ATP. It was also coming up with ways to store that ATP, right? So that that glucose can then be broken down again when needed, right? Also, I mentioned this to you last time, right? Different parts of a tree, the leaves are photosynthetic, so all the cells in the leaves get plenty of ATP just from doing photosynthesis, right? But what about the cells that are in the branches or the trunks or the roots of the tree, right? They're not doing photosynthesis, but they need energy as well. So that's the other reason plants evolved to, or photosynthetic organisms, right, evolved to make glucose, because glucose can be transported throughout the tree down to the roots. And then the cells of the roots can take the glucose in, right, and break it down to get the ATP back out of it again, right? Okay, so how does the plant take some of this ATP that's being made in the first part of photosynthesis and convert it into sugars? That is the second part, right? But first, I want to, before we get to the second part, I want to zoom in on this electron transport chain and look at it in a little bit more detail. All right, I kind of got ahead of myself here. All right, so let's look at this. So here is photosystem 2. This is the one that we looked at last time already, right? We said it kind of looks like a big purple bell pepper, right? And so it has a core part of it that has that special P680 chlorophyll, and it's surrounded by other protein subunits, many protein subunits that are kind of holding on to these chlorophyll A's and B's, right? So these are these different sort of photosensitive pigment molecules that are essentially being held onto by other proteins as sort of prosthetic groups, right? These are like the equivalent of the baseball gloves in the example of the mitochondrial electron transport chain that we discussed earlier. Right? So once again, the photons of light, they excite the electrons. You get this resonance energy of different chlorophyll molecules kind of crashing into each other, and that's how the energy is kind of passed along. Finally, they crash into this central P680 chlorophyll, causing it to cough up a pair of electrons, right? And that pair of electrons is then carried off by this special pigment molecule, this plastoquinone, or QB. And again, right, the electrons that are coughed up by this P680 have to be immediately replaced. And those electrons come from the water molecule being broken here to get those two electrons back. And that's where the oxygen comes from. So the H2O is broken to return electrons to the special P680 chlorophyll so that it can continue to donate these electrons to the electron transport chain. Right? So surrounding these photosystems are other proteins that are just kind of in the membrane that are just loaded up with all kinds of chlorophyll molecules. And because these are kind of hovering around or floating around the photosystems, they're kind of free to congregate and become more closely packed together, or they're free to kind of space out, right, and get farther apart. So look at this figure here. So in this figure, these gray blobs represent this photosystem 2 core, right, with the P680, and all of these separate little green things are these light harvesting complexes, or LHCs, that are little proteins holding on to these chlorophyll molecules, right? And they can all sort of collect around the photosystems and are free to move around in the membrane. So in low light conditions, these light harvesting complexes really pack together. So by packing together, whatever available photon energy is hitting them is more efficiently transferred through resonance energy, right, through these different chlorophyll molecules to kind of encourage more of that energy actually making it into the core of the photosystem. So I think hopefully that makes sense to you, right? So I think these little yellow, I don't know what these are, little marks here are meant to represent the fact that these two are close enough they can connect. So as energy is being sort of bouncing around in the double and single bonds of this one, it can actually crash up against this photosystem or this light harvesting complex over here, and that energy can then be transmitted, right? So that's under low light. Under bright light conditions, remember, there's the risk of sort of overtaxing or overloading the photosystem, right? Getting electrons flying off of the P680 chlorophylls faster than they can be replaced, which would be really bad. So in that type of a case, you can see not only do these light harvesting complexes tend to be more grouped farther away from the photosystem cores, but you see the location of various accessory pigments like the xanthophylls, right, that are positioned here to be able to help absorb some of that extra energy so that it won't overload the PS or the photosystem cores here, the gray blobs, right? I think that's kind of cool, right? Because you can imagine over time this happening, because again, all of these proteins are just anchored into the membrane, right? But they're not actually like anchored right in place next to each other. They're free to float around in the membrane. And so they can spread apart or come together in order to respond to changing light conditions. So this big gray blob here, right? So this big gray blob is a more accurate representation of the shape of this big purple bell pepper on the left here, right? You might say, well, what's that about? Because I really want to see what this blob looks like, right? So this blob, let's take a closer look at it. Right, so here it is. So this is the actual structure, the three-dimensional space-filling structure, right, of photosystem 2. So it's made up of many subunits of proteins. Again, another wonderful example of what we've been learning about in this class, that individual proteins, right, that are each coded for by different genes of the plant, right, those proteins are meant to fit together to form large, complicated machines, right? And this is a massive machine here, right? And you can, so this is all the exterior surface of all the electron orbitals of all the amino acid side chains sticking out, right? I mean, this thing is huge. And when you look at this, I kind of imagine this as being like this huge kind of like thing that if you were, you know, approaching it, if you were teeny tiny, right, like in a little, if this was a massive, I don't know, space station or something, and you're like a little spaceship flying up to it, right? It'd be something like you'd see in Star Wars, right? But you say, okay, well, I get it, right? Let's see what this actually looks like on the inside, right? Different ways of looking at protein structure. Right? So on the inside, you can see all the different peptide chains, right, that represent these different proteins. So each different colored peptide chain here is a different protein subunit. So you can see all of them working together, right, to make the correct shape. And we see secondary structures like these helical structures or these different beta sheets, right? We see some parts of the protein peptide chains are sort of unstructured, right? So here the peptide chains are kind of more free to kind of move around. And what are these little green, little zigzaggy things in here? Those are chlorophylls, right? Those are chlorophyll A's and B's that are being held onto by these proteins. So if this is the big purple bell pepper, right, this would have been the little green circles in the cartoon in the bell pepper, right? So energy from other light harvesting complexes that are floating by, right, would crash into these, and then these chlorophylls then would continue the process of transmitting that resonance energy of the electrons, eventually reaching, I think it's this part of the complex over here, where you'd have special P680 chlorophylls that would be the ones that actually cough up their electrons into the electron transport chain, and then water down here would be broken, right, to replenish the electrons so those P680s can keep working. Right. How cool is that? I think that's pretty amazing. Okay, so this is the rest of the electron transport system then. So again, here's photosystem 2 that we've been looking at. So the electrons that are coughed up, a pair of electrons coughed up by the P680 are taken by plastoquinone, so QB, right? So plastoquinone then has a pair of electrons that it's carrying as it comes out of the photosystem, right, it wants to be able to, so if it has these two negatively charged electrons, it would love it, right, to be able to pair these up with a couple of protons to kind of neutralize that charge. So protons from this side of the membrane get paired up with the electrons. And so that means the plastic quinone is now reduced, right? It has two extra electrons on it with protons. And so that reduced form, we can just call QBH2, right? Because it has these two, essentially two hydrogen groups attached to it. That reduced plastic quinol goes into this complex here, this cytochrome BF complex, right? Which essentially removes the protons from the electrons that were being carried by the plastic quinol, right? And the way it removes it is it ends up releasing those protons on this side of the membrane, right? So that's how it's actually pumping these protons to the other side. The electrons then are received by this plastocyanin molecule, this PC, and then carried to photosystem 1. Remember, the photosystems are actually labeled photosystem 1 and 2, even though the order that they sort of participate in this are sort of out of order, right? It kind of starts with photosystem 2 and eventually feeds into photosystem 1. Those electrons then that are brought in from the plastocyanin, right, they can replenish electrons that are being booted off of this special chlorophyll molecule, P700. And what's booting the electrons off of this one? Well, more photons, photons of light coming in and exciting the chlorophyll in the photosystem 1. That's what's causing electrons to be booted off of the P680, right? And so they're being replenished by these electrons coming in over here. This pair of electrons is carried off by ferredoxin, or FD, right? And so carries the electrons out. And it's carried by this enzyme, ferredoxin NADP reductase. Right? So I know you're already like, oh boy, right? This list of names just keeps getting longer and longer and longer. But, you know, think about the name. What does it do? Ferredoxin NADP reductase, right, is an enzyme, because the ACE part of it tells you that, right? And it's going to work on ferredoxin and NADP. Right? So here's NADP, another variation of these electron carrying baskets, right? So it's a nicotinamide adenine dinucleotide with a phosphate already part of it. It is positively charged. It can join up with another proton on this side, right? So it actually kind of has two positive charges. You can think about it that way, right? So this FNR enzyme will catalyze the transfer of those electrons that are coming out of the electron transport chain onto this NADP molecule. So you end up with NADPH, right, carrying these two electrons. So that represents the terminal electron acceptor of the photosynthetic electron transport chain. Remember, in the mitochondrial electron transport chain, what's the terminal electron acceptor that's so important in order to keep it running? It's oxygen, O2. Right? So the O2 that we breathe in receives the electrons at the end of the mitochondrial ETS chain, right? And it becomes water, right? And that's the end of the story for those electrons. But in photosynthesis, right, these electrons can still be useful for another important function. And that's why, rather than this ending up with oxygen again, just receiving them and turning it into water, right, in this case, this electron carrying coenzyme, NADPH, will carry these electrons off to the Calvin cycle. Right? The nice thing about this system is if the plant runs into trouble, meaning if the plant really needs to make more ATPs, and that's the focus, right? It's like running short on ATPs, then rather than shipping these electrons off somewhere else, these electrons can actually be recycled. So this FNR enzyme, rather than filling this electron carrying basket up here to send those electrons away somewhere else, this FNR enzyme, if needed, can actually feed those electrons back into the cytochrome BF part of this pathway in order to encourage more protons to be pumped so that the ATPase proteins that rely upon the protons, right, have plenty of protons to be able to drive ATP production. All right. Okay, so those electrons that are being brought out of the photosynthetic electron transport chain by this NADPH basket, right? That feeds into the Calvin cycle down here. So this is the second part of photosynthesis, carbon assimilation. Right? This is where you're going to take some of the ATP that you've made from energy transduction part of photosynthesis, right, and convert them into an organic molecule, right, into carbon-carbon bonds, right, that can then represent a way for this energy to be shipped to other parts of the plant, and they represent a way to sort of save this energy for a rainy day, literally, right, a rainy, cloudy day when you don't have enough sun to continuously make ATP on this top part. Right? So we call this carbon assimilation. What does it mean? It means you're taking carbon atoms and assimilating, right, or joining them with existing organic molecules to make the particular carbohydrate or other organic molecule that you need to make. So that's why it's the second part of photosynthesis that actually involves the CO2 coming in, right, and using some of that ATP that was made from the first part to go ahead and make sugars. So let's look at this Calvin cycle. So the Calvin cycle involves, right, the fixation of atmospheric carbon. So carbon that's floating around as carbon dioxide, right, has to be taken by the plant. That carbon is taken off the CO2 and it is fixated, right? It's actually put as part of an organic molecule, right? That's what we mean by fixation or assimilation. So the Calvin cycle has 3 stages. First is the carbon from the CO2 in the atmosphere is going to be added to an intermediate molecule, which is a ribulose sugar. And that's going to serve as the acceptor that takes the carbon. Then the second stage is going to be taking electrons from the electron carrying basket, the NADPH that came from the first part of photosynthesis, right, the leftovers from the electron transport chain, right, and reducing that acceptor molecule to make glyceraldehyde 3-phosphate. And you're like, glyceraldehyde 3-phosphate, G3P, right? Where have I heard this before? That's the same intermediate molecule, the G3P, that's part of glycolysis, right? So you can already see, oh, already it's connecting, right, with other pathways that have to do with the management of sugar. Right? And then the third part of the Calvin cycle is to regenerate the original ribulose molecule that accepted the carbon in the first place so that this cycle can keep going around and around. So here's the ribulose, here's ribose sugar, right? So this is the same ribose that's in our nucleic acids, our nucleotides, like RNA, right? So instead of being a ring, it's just shown as a linear structure here. So it's the 5-carbon ribose sugar. So ribulose is a variation of that. You can see carbon 1 and 2, right, are different. So this is ribulose. So this is the Calvin cycle. Again, this looks scary, but I don't expect you guys to memorize these structures, but it helps when you're studying to look at these structures. So that way you can kind of figure out what's going on. Here's the ribulose 1,5-bisphosphate, ribulose that has these 2 phosphate groups. Notice the ribulose is 5 carbons. So most of the intermediate molecules that have to do with sugars, especially in glycolysis, how many carbons were those? Remember, those are like 3 carbons, right? Because the 6-carbon sugar as part of glycolysis gets broken down into 2 3-carbon intermediates. Right? And that's true all the way down to the level of pyruvate. Right? So if you're starting with a 5-carbon molecule, you need to add 1 more carbon to have 6 so that you could then split this thing into 2 3-carbon intermediates, and each of those can be worked on, right, and eventually made together to form a 6-carbon glucose. So that's where the carbons come from. So if you have 3 ribulose 1,5-bisphosphates, so 3 of these 5-carbon molecules here, and you introduce 3 molecules of carbon dioxide, right, that means you have 3 carbons. Each one can be added to one of these 5-carbon chains to make a 6-carbon chain, right? And then that 6-carbon chain can then be split in half, right, to create 6 of these 3-phosphoglycerates, right? Then the second stage is taking this 3-carbon intermediate and turning it into glyceraldehyde 3-phosphate, G3P. So this involves, the electrons from the NADPH that came from the first part of photosynthesis coming in here to kind of reduce this molecule and create glyceraldehyde, or G3P. Right? So of these 6 molecules that are made, 5 of them are going to continue on and be restored back into ribulose. But 1 out of the 6 of these gets funneled off to make things like organic molecules, sucrose, starch, and other molecules. Okay. So what do I want you to know from this? I want you to know the 3 stages in terms of, just thematically what's happening there. You don't have to be able to draw these structures, but again, when you study this, the structures help you kind of understand it, right? I want you to understand what they do. I want you to understand that you're going from ribulose 1,5-bisphosphate to glyceraldehyde 3-phosphate, and this glyceraldehyde 3-phosphate can then be fed into different sugar pathways, right? And then some of this G3P is restored, regenerated back into ribulose 3-bisphosphate. So a very important molecule here is this first enzyme, this Calvin cycle 1 enzyme that actually takes the carbon from carbon dioxide, right, and puts it onto this acceptor molecule. Right? So this enzyme is called Rubisco. And so Rubisco catalyzes carbon dioxide assimilation into making these triose molecules, these 3 carbon molecules, right? So again, 3 carbons from the atmosphere are brought in, joined up with 3 molecules of ribulose 1,5-bisphosphate. And so then you end up with the ability then, each one of these carbons will be added on to one of these. You have 3 6-carbon molecules that can then be divided, split down the middle to make 6 of these phosphoglycerates that then get converted into glyceraldehyde 3-phosphate, right? And then sent off. One of these 6 are sent off to be made into sugars, right? So the amazing thing about Rubisco is all photosynthetic life on Earth has Rubisco, right? Because it's such a fundamental, important step here, is taking the carbon dioxide from the atmosphere, right? So all life, photosynthetic life on Earth has Rubisco. Right? As a result, Rubisco is actually the most abundant protein on Earth, right? I remember reading somewhere that if you were to took all the Rubisco on this planet from all organisms that photosynthesize, right? I cannot even remember. It was something ridiculous, like how many hundreds of tons of this protein you would actually have represented by life on Earth, right? So it's really fascinating to think about, because the Rubisco is positioned right here. It takes care on the right here of taking the carbon from the carbon dioxide and sticking it onto the ribulose 1,5-bisphosphate, eventually leading to the ability, right, to break this into 2, 3 carbon groups that can be worked on further as the building blocks to then make a 6-carbon glucose. Right? All right, so last slide for today, just sort of a summary. Again, right, why do plants need to make sugars in the first place? Because photosynthesis is able to convert 26 photons of light into 9 ATP molecules, right? However, many of the cells in a plant don't have access to the sunlight directly. So that means many of the cells in the plant actually depend upon, right, that energy that's being transduced or converted into chemical form to be transported within the plant so they can get some of that. Right? So that's why it needs to be made into glucose. Consider also at night, there's no light for photosynthesis. So what happens at night then if there's no photosynthesis? That means the plants have to rely solely on taking that glucose that it's made and doing aerobic respiration on it in order to break the glucose back down to get the ATP back, right? The same aerobic respiration that our bodies are designed to do. That's why, again, right, plant cells absolutely have mitochondria in them, right? Because the mitochondria is what's going to take care of doing aerobic respiration at nighttime. So at night, right, it's a very different story than during the day. During the day, when there's lots of sunlight, carbon dioxide is taken in by the leaves of the plant, O2 is released, but at night, it flips around, right? Oxygen is taken in by the plants to do aerobic respiration, and carbon dioxide is released, right? And we depend upon this glucose that plants make, because animals that eat the plant to get the glucose, other animals eat those animals so that the energy gets passed on to them. And then we come along and we either eat the plant directly or we eat the animal, right? And so that energy then becomes part of our body in the form of glucose and other nutrients, right? But that energy and the glucose originated from the sun. And that's why we are all inexorably linked. All life on Earth is linked, whether it is photosynthetic or heterotrophic like us. All right, let's go ahead and stop there.